Kishore and we overlap. So we are the same batch.
Okay, go ahead.
72 to
Just let
77.
me know if you can hear me.
Yeah, clear.
Okay.
And then I am sharing my screen.
We can see the screen.
Okay.
Yeah, we
perfect.
could, yeah.
Yeah, thank you very much, Dr. Chakramesh and all the IISc alumni team for giving me this opportunity. I was in IISc from 91 to 97. Right now, I am in University of Alabama at Birmingham, which is in a deep South state. I am part of a cancer center here and also Department of Pathology. I still have my roots at the University of Michigan and Kasturba Medical College in Manipal, so I am an adjunct faculty there. Of course, I have no financial disclosures. I come from Udupi, which is in the west coast of Karnataka, basically a small town near Udupi called Brahmavara. And I did my undergrad studies in Purna Prajna College, which is education from Admar Mutt, the 8 mattas of Udupi. One of the matta is Adamar Matta. They run this education institution. After my BSc, I went to this medical college there to do my master's in clinical biochemistry. This is the college that's set up by Dr. TMA Pai and is running from last over 60 years, I think now. They have divisions all over the country now and also in some foreign countries. Of course, then I went to IISc Biochemistry Department to do Ph.D. with Dr. Bldg and then mentored by Dr. Vishwesharaya as well. If people didn't go to IISc nowadays, this building, I don't think many people occupy this building anymore. This is the biochemistry, and all the faculty went to the new building there near the D gate near Mattikere. After my PhD with Dr. Bldg and Vishwesharaya, I moved to work as a postdoc with Srinivas Kaveri in Paris. And right now, Srinivas Kaveri is a liaison officer for Indo-French science, I think, collaboration. So I was with him for a couple of years, working in the basic areas of immunology. And after that, I went to University of Michigan again to do another postdoc, and I switched from immunology to cancer biology. And I was trained by Dr. Arul Chinayan, who is a Howard Hughes investigator now. So after my postdoc there, I became a faculty there itself, stayed there till 2015, and then moved south, where weather is much better in terms of cold. And I'm here from last almost 10 years. And all from 2000 when I joined Arul, and now till now I'm working in cancer genomics area and target discovery. So I'll give a very basics of what we do and about cancer as well. So in simple terms, cancer is a disease which, you all know, the cells grow uncontrollably, and during progression, some of the cells may go into the blood, and the cancer cells may go to distant organs and cause metastatic disease. And my goal from last 25 years is to identify some of the targets that change in cancer, and that can be either used as a biomarker for early detection of different cancers, or use them as a target for inhibition by small molecule inhibitors. So as we know, during the differentiation of the fertilized eggs, different cells become differentiated into different organs, and all this change happens because of the gene expression changes that occurs. And for example, muscle cells will start expressing muscle-specific genes, and they don't express other genes. If you consider there are about 20,000 genes in each cells, some of the genes that is required for muscle cells may be expressed, not the other cells, right? So similarly, different organs will express different genes, and this expression is maintained throughout the course of that lifespan. And all this message comes from DNA and then transcribed into RNA and translated into protein and all that. So this is maintained, like I mentioned. Let me see if I can show. This gene expressions is maintained once the cells are differentiated. They start expressing certain genes, not expressing other genes like that. But when that program is messed up, you start seeing differential gene expression, which is not common to that organ, and that many times can lead to cancer. So in last 20-25 years, we could measure all the genes in any organ or any cells using high throughput technologies. Like earlier it was microarray technology. Now we do the next generation sequencing of DNA, RNA, and all that. And we use something called heat map. We all seen this probably, where you can look at all the genes and across different cancer patients. And you can represent with red signal as overexpressed genes and green as underexpressed genes. So the molecular identity of these cells need to be maintained during, in the organs. And if that is messed up, you start seeing different gene expression. And that we can measure nowadays across 20,000 genes. The cancer itself is a complex disease, as we all know. This slide, as you can see, early stage, there may be few cancer cells, and they may accumulate different mutations and gene expression changes. And over its course, it started accumulating additional changes, start growing faster, develop blood vessels, and becomes very aggressive and invasive cancer. And the cancer of different organs have a different time course. And in individuals also, the cancer may grow fast or may take years together. In many cases, for example, in prostate cancer, many men die with prostate cancer, not because of prostate cancer. So those are all the extreme cases. But most of the time, if you can detect cancer when it is in the early stages, surgeons generally try to remove the cancer spot, or if it is already late stage, they start treating with either chemotherapy or targeted therapies. And the cancer, individual organ cancer itself is very complex. This slide very nicely illustrates all this histological photomicrograph you see are from breast cancer, from different patients. But what you do see is it's very showing different patterns. And when one sequences all this different type of breast cancer, they find different mutations or gene expression alterations. And nowadays, there are many drugs that are developed specifically targeting these mutations. So if you're treating patients, you want to have all the information that is available now, histological, morphological, and gene expression, as well as mutation profile of the individual patients. Of course, the cancers also, like I mentioned, during late stages, cells can move out of the cancer, the original cancer location, and get into the bloodstream, go to the distant organ and start growing there, causing metastasis, which even now almost always lethal, and patient will die because of this metastasis. Like I said, in the last 20-25 years, we have different high throughput technologies that are developed where you can look at individual patients, every nucleotides, the genome, the transcriptome, and the epigenetic changes like methylation, promoter methylation, and you can look at transcription factor bindings, as well as you can look at genes that are not coding for proteins like small RNA, like microRNA, or long non-coding RNA as well. And also mass spec technology allows, these days to look at all the protein expression as well as post-translation modification. So you can use this technology to see what is the difference between normal tissues or cells versus cancer. Of course, you can use mass spectrometry also to look at all the metabolites. So this is the genesis of Omic technologies, where you would measure all the genome or transcriptome changes, proteome or metabolomic changes. And we can use all this available information to find what changes in individual patients. And that generated a concept called precision medicine or precision targeting, particularly in cancer. In almost in every hospital in USA, any cancer patient comes in, they will be sequenced, particularly nowadays about 350 to 500 genes they will sequence to identify particularly mutations. And there will be tumor board that will analyze, which consists of pathologists, surgeons, oncologists, bioinformaticians, and see if there is a avenue to target that specific individual patient with the available drug. Of course, the precision targeting is enhanced because of all these drugs they are developing based on identifying mutations in specific genes particularly, and then developing therapeutics for them. So this slide shows the targeted therapy for different mutations in different cancers, basically. However, while the precision targeting is a great concept, there is still a lot of issues. Particularly, this slide basically shows around 750 patients were sequenced to identify targetable alterations, and at the end of the day, they could target only 16 patients. That's purely because there are not enough targets, and there are not enough targets that are targetable with available FDA-approved drugs. As far as scientists, we also have other issues, like we don't want to, or we can't go to some new targets to work on, because if you submit NIH grant, they will say, what is this new molecule and what is the guarantee it will work? So people always go after P53, EGFR, or ERB2, those kind of genes. But there is a change happening, and a lot of new data, genomic data, is helping in identifying new targets as well. So I started my cancer research when I joined the University of Michigan by doing microarray analysis of gene expression changes in prostate cancer with ARO. So we collected RNA from benign prostate and early stage prostate cancers, and also metastatic prostate cancer. Prostate itself is an organ that's present only in men, and it is regulated by a hormone called androgen. Okay, so we collected all this RNA from different tissues, hundreds of patients, and did the microarray analysis, and identified many genes that are going up or down in prostate cancer. So as you can see in this slide, from normal prostate epithelium, as the cancer progresses, we see different genes either up or down regulated, different classes of genes. And then we went on to validate some of these genes and try to understand the biology of some of these genes. Validation involves the protein expression analysis using hundreds of patient tissues, where we generate something called tissue microarray, where we put hundreds of individual patient tissues on a slide and immunostain with specific antibodies and see if proteins also changes along with the gene expression. Most of the time we analyzed, around 75% of the time, if there is increased gene expression, we do see increased protein expression, but there is discordance as well. So meantime, the NCI had initiated a project which is very well known now. It's called the Cancer Genome Atlas, where they collected sequencing information from over 10,000 patients. That information includes the DNA sequencing information, RNA sequencing information, as well as some of the epigenetic changes like DNA promoter methylation alterations. And later, they also did the clinical proteomic analysis, post-translational modification analysis and all that. And they deposited this data for anybody to use across the world, basically. The bottleneck was how to analyze and use it by common cancer biologists or researchers. So that is when we decided to develop a tool that anybody across the world can use. So of course, there is huge amount of data, so we downloaded all the data and put it on our own servers here, and then started analyzing the data. So the proteomic data was analyzed by my collaborator, Chad Creighton at Baylor, and the transcriptomic data was analyzed by Darshan. And once the analysis done, we developed a platform, a web-based platform that anybody can use. And we called it You All Can, basically a simple tool so that anybody can use it. I'll go through a little bit of the utility of this tool, and of course, the detail about how we developed and what we can do with this tool are there in all our published articles. The tool itself, if you Google You All Can, you'll find it. And also we now have You All Can mobile app where you can download the app in Androids or iOS. Web-based is a very large platform. And you see this kind of page where you have tutorial as well if you want to analyze. And then we have TCGA, the transcriptome data, the clinical proteomic data, and pediatric brain tumor data. So once you click the button, let us say TCGA, you get into this page, and where you have on the left-hand side different cancers with the downward arrow, because we subclassify the cancers based on the information available from the TCGA, which may include histological subtype, molecular subtypes of that specific cancer. Or if you want, you can enter the gene of your interest and select cancer of your interest here. And in the top, you see there is a gene expression, microRNA expression, link RNA or long non-coding RNA expression, and prognostic biomarker buttons. You can click any of them. So then you select any of the cancer. For example, here I wanted to look at gene expression of prostate cancer, particularly a molecular subtype of prostate cancer called X gene fusion positive prostate cancer. Then we get this heat map. When you click here, you get something called heat map, where each line here is an individual patient. And these are all the top overexpressed genes, basically. So you have normal prostate RNA expression, and then there is the class called X gene fusion prostate cancer, and then prostate cancer without that X gene fusion. So you do see some differences between the X gene fusion versus non-X. So you can click on any gene and start further analysis. And you can download all these images from You All Can using this button here. You can download as SVG or PDF or JPEGs, and you can edit the font, etc. For manuscripts or grant proposals. So once you start the analysis, you can start looking at different subtypes of expression based on different subtype of individual cancers. So all this subtype information comes from TCGA, and we look at the gene expression based on these subtypes. So for example, there is a gene here called PLA2G2F. It is expressed only on the luminal subtype of bladder cancers, not the other common subtype of bladder cancer called basal subtype. So this gene is not expressed in basal subtype, only in luminal subtype. So assuming there is a drug for this gene, you want to select only this luminal subtype of bladder cancer for treatment, not the basal subtype. That is the advantage of classifying cancer patients based on various factors, right? And of course, you can also look at the survival of survival information of patients based on increased or decreased expression of specific genes. So this is called Kaplan-Meier survival analysis. So for example, here, this gene NPEPL1, if it is high in renal cancer, the patient dies very faster. This kind of information can be used theoretically for treatment purpose. Looking at this gene, if it is high, the clinician can go for aggressive treatment because the patient dies faster. You can also look at these buttons here in the You All Can and do further different analysis. For example, you can look at correlation analysis, or how your gene of interest is correlated to other genes. For example, here, lamin 1B gene is very highly correlated with CDC25C. This gives for biologists some information. Maybe there is a interregulation between these genes. Maybe CDC25C is positively regulating lamin 1B. You can always go to lab and knock down one or the other gene and see what happens if there is any change in the expression pattern, and then further validate the biology. And one of the important thing also, at least in US, they are trying to do is see gene expression changes based on different races, because one of the things they commonly see here is that African American patients are having generally aggressive cancers, and most of the time, they die faster. That could be simply because of socioeconomic status as well, but still there is some differences at the gene expression level that one can identify. TCGA does not have too many African American patient samples, but whatever is there in certain specific cancer, we try to provide information. Talking about that now, in India, they have started a consortium called Indian Cancer Genome Atlas. Anand Deshpande is spearheading that effort, and they're trying to start with breast cancer sequencing. Idea is to generate that data and start putting it out for public use. So I'm in touch with them if they generate those data, and we'll try to incorporate that into UALCAN as well. And you look at any of the genes, you see this button, buttons for different analysis. You also see external links. This is the idea of putting this external link is basically to help researchers quickly understand about that gene and what is known about that gene, and if there is any drug target. For example, we provide information about any drugs that is available. It may not be FDA-approved drugs, but that drugs that may work on that gene. So all this information PubMed we provide, because how much is known about this gene, either in cancer or in general in biology. So we give all this external links as well. Some of the use of external links, for example, the Human Protein Atlas is basically a immunohistochemistry database across 16 type of cancers, where they did immunohistochemistry on these patient tissues and provided the images, basically. For example, here there is a gene called GLY8AL1. When you look at different cancers in UALCAN, you see prostate cancer specifically has higher expression. Red is cancer and blue is normal. You see higher expression of this gene in prostate cancer. Then if you go and click the Human Protein Atlas, you get this information, and you do see the immunohistochemistry, that brown color there, specifically staining prostate with this antibody GLY8AL1. And if you click on that, you get a very high resolution image of prostate cancer tissues, and you can see that there is benign glands, prostate glands, versus cancer. So you can find that this not only gene, the protein is also increased in prostate cancer. So you can look at around about 15,000 antibodies that they may have in this database. And many times the antibody may not be the greatest antibody, but some of them are very good antibodies, and you get very high resolution images, so that you can go back to lab and you can look at your own patient tissue cohort and do the immunostaining. And there is information that we provide from TCGA called DNA promoter methylation. This is an epigenetic change, as many of you may know. So if there is high or increased promoter methylation of a specific gene, it generally represses the gene expression or silences the gene expression. And many tumor suppressor genes are known to be promoter hypermethylated. And when you look at that, for example, this gene, SOS-TDC1, is hypermethylated in prostate cancer. And if you ask for gene expression, it shows it's downregulated in prostate cancer. And we also provide a lot of proteomic information that comes from Clinical Proteomic Consortium. You can look at the protein expression changes across cancer and again do all the analysis that we provide with the gene expression, basically. There are genes that don't code for protein. They may not have open reading frames. Yes, the long RNAs that don't code for protein called long non-coding RNAs, we provide information, differential gene expression for that as well here, as well as the small non-coding RNA called microRNAs. MicroRNAs are known to negatively regulate gene expression by binding to 3' UTR and regulate the gene expression. Just give quickly a couple of examples how you can use UALCAN to find something that may be novel and useful in cancer research. So we were looking for genes because I was working extensively on prostate cancer. We also identified there are gene fusions in prostate cancer when I was at Michigan. But gene fusion is basically X transcription factor fusions. So they are hard to target. It's a nuclear protein. So I was looking at ERK gene fusion, positive prostate cancers where you have ERK here and they have gene fusions. I was looking the correlated genes along with the ERK, and you see all these genes potentially maybe co-expressed with ERK, not only co-expressed, may be regulated by ERK. So among that genes, we found that frizzle date is also increased. Frizzle date is a Wnt pathway receptor gene, okay? So Wnt pathway is extensively studied. There are many experimental drugs for the whole pathway itself. So if at all some of these drugs becomes available, FDA approved, we can think of potentially targeting prostate cancer with ERK gene fusion with frizzle date inhibitors. As you can see, when you subclassify based on different molecular subtype, the prostate cancer, again, you can use UALCAN to do that. You see that ERK is expressed only in this gene fusion positive prostate cancers. You have a number of patients is 152. None of the other prostate cancer had this gene expression. And when you look at the frizzle date, which is a Wnt pathway receptor, it is also expressed mainly or only in ERK gene fusion positive prostate cancer. So what is this gene fusion? Basically, I mentioned prostate is an androgen regulated gene. And in normal prostate, you don't have this fusion between TMPRSS2, which is an androgen regulated gene, with this transcription factor. But around 50% of prostate cancer have promoter of TMPRSS2, again, which is an androgen regulated gene, fused with ERK transcription factor, causing this gene fusion. So when there is androgen, dihydrotestosterone binds to androgen receptor, they enter the nucleus, sit on the TMPRSS2 promoter, which is now fused with the ERK, and drive the ERK gene fusion, causing oncogenic gene fusion. What we showed here now is that that ERK itself in turn drives the expression of frizzle date. And as I mentioned, and of course, we did a lot of validation to confirm that ERK drives frizzle date expression. For example, this immunohistochemistry shows that when there is ERK gene fusion in prostate cancer, they express high level of frizzle date, whereas non-ERK gene fusion, which has other molecular biomarkers like SPINK1 positive prostate cancer, don't have frizzle date expression. We also did chip analysis to show ERK binds to the promoter of frizzle date, and also did the knockdown studies. So why we did all this is because there is a lot of drugs, like I mentioned, that they're trying to target Wnt pathway. So eventually, if any of these drugs that is listed here becomes approved for inhibition of Wnt pathway, we can think of targeting those ERK gene fusion positive prostate cancer with these drugs. There may be other drugs that are available in different pharmaceuticals that failed clinical trials as well. Many of you may have worked in pharma as well. Some of the reason why the clinical trial may have failed is purely because the the patient population they took. For example, here is the gene called IDO1. IDO1 is a tryptophan metabolizing enzyme. And if you see in bladder cancer, for example, slightly increased in bladder cancer, but when you subclassify bladder cancer gene expression for IDO1, you see only basal squamous type of bladder cancer have IDO1 expression. So if the clinical trial, which is generally set up across patient population, it may not really work well because many of the patients will not have the target basically. So now, if of course pharma want to target big patient population to recoup the money they spent on developing drugs, but idea of precision medicine is even if there is a small patient population, you should be able to use the available drugs. There is cost involved, the clinical trial costs and drug development cost and all. So that is something you can't avoid basically. But still, if there are drugs that failed clinical trials, now it is a time probably to get back them and look at the patient population, stratify them based on all this information that is available now, the sequencing information or proteomic information, and then potentially set up clinical trial again with a select patient population. Of course, you all can, we released it about 7 years back. And of course, we did a lot of additions and new graphics, new analysis, new data sets we included. And it has been extensively used across the academics as well as pharma. This is a nice, positive words from Jay Bradner, who was at Novartis. And of course, we also have information about how many people use every day and across the world. I'm very happy to see people from India, a lot of them also using this tool, and in fact, a lot of use from US across different cities. Like I mentioned, we also developed a light version of ULCAN called ULCAN Mobile. This is a mobile application because if somebody is in the grant review committee or sitting in the airport and they like to look at genes, they can download this app and look at gene expression changes across different cancers, basically. So I know we also have all these targets, but how you go about validating some of them? I give some information about this gene we worked from last 20 years. It's a gene called EZH2, which is a histone methyltransferase. So we found this as a gene that is overexpressed in prostate cancer, and we did not know much about that during those days. So Arul said, select some genes based on certain criteria, it should be in aggressive cancers and start validating. So one of the gene I picked up was EZH2 without knowing much about it. And we did validation, particularly the protein expression validation, and we found that it's very high in aggressive prostate tumor as well as metastatic prostate tumors. We analyzed hundreds of patient samples by this tissue microarray technology. What was interesting for us was that if there is EZH2 is very high, patient die faster. Or in this case, specifically in prostate cancer, the PSA, the prostate-specific antigen, comes back. That indicates that it's cancer cells growing that secretes PSA. Similar experiments we did in breast cancer as well, and we found that EZH2 is very high in breast cancer and patients die faster in breast cancer. And later, of course, it has been identified in many solid tumors as very highly expressed, particularly in aggressive tumors, and about 25% of lymphomas, DLBCL class of lymphomas, have activating mutation in this gene. So one of the things we needed to do that time was to test if the EZH2 is oncogenic. So we could add the gene into normal cells and check for some of the assays we do in the lab, like colony formation, invasion, tumor growth, or we can remove EZH2 by siRNA technology and see what happens to the cancer phenotype, like cell proliferation or colony formation. So here we show that in normal transformed breast cells, if you add EZH2, it starts forming colonies. We also showed it increases invasion. If you remove EZH2 from aggressive cancer cells, it blocks tumor growth. So we wanted to check what is EZH2 doing. So we put EZH2 into normal cells and did a gene expression analysis. So what we found was that if there is EZH2, a lot of genes are decreased in those cells. Later, Yi Zhang, that time at University of North Carolina, showed that it's a histone methyl transferase. What it does is adds 3 methyl groups to lysine 27 of histone tail H3. So what it means is that if there is EZH2, a lot of genes are downregulated. And when we started analyzing, most of these genes are tumor suppressors as well. And also their promoter is also hypermethylated. So it suggested to us that EZH2 somehow, because of this methylation, blocks expression of tumor suppressors. This is a nice cartoon showing what happens is EZH2 adds 3 methyl groups, then compacts the chromatin. And that prevents transcription factor from binding to the promoters. That is how it blocks the RNA expression and protein expression. And incidentally, many of those genes also have promoters that are methylated. So there is a double dose of silencing of these tumor suppressor genes when there is EZH2. We validated some of these tumor suppressors. One of classic example is this E-cadherin, which is a cell addition molecule, makes cells attach to each other. Here, the red color is E-cadherin. So when we add this EZH2, which is a nuclear protein, you see the green color, the E-cadherin start disappearing. And this is how we believe EZH2 prevents cells from attaching, makes the cell loose and make them invade, causing metastasis, basically. Of course, we went on to extensively understand the mechanism of EZH2 function as well as regulation. One of the reason EZH2 is increased in many solid tumors is because it is regulated by a microRNA called microRNA 101. And microRNA 101 is downregulated in tumors. Because of that, EZH2 goes up. We did a lot of functional study to show this. So these are all the experiments we do when we have a target in the lab to show why it's important, how it functions in cancer. With the goal of course, targeting them eventually, either in our labs or somebody in the other labs or pharmaceuticals may pick this up. So we started screening some small molecule for EZH2 because it's a enzyme and also it regulates tumor suppressor, downregulates. So our idea was if you add inhibitor, the tumor suppressor should come back up if it blocks EZH2. So that is what we did. We knew that EZH2 causes downregulation of E-cadherin here, the black box. And when we add our small molecule or even HDAC inhibitor, the E-cadherin expression comes back up at the RNA level as well as the protein level. You see the E-cadherin protein is downregulated when we overexpress EZH2 in cells, normal cells. And when we add our inhibitors, the E-cadherin comes back up, showing that this molecule may be useful to block EZH2 functions, basically. We used this small molecule in animal models as well. We did see some downregulation of gene expression. We didn't go too much further because by then, a lot of pharmaceuticals developed very highly specific EZH2 targeting inhibitors. So because a lot of biochemists are there, EZH2 basically trimethylates lysine 27. For that methylation, it needs methyl group, and the methyl group donor is S-adenosyl methionine, SAM, right? So these small molecules are basically analogs of SAM, S-adenosyl methionine. So they block EZH2 using SAM, basically. So that is how they inhibit. All these specific inhibitors inhibit. The GSK developed it, Epizyme developed its own inhibitors. And many of them were used in clinical trials, and now some of them are FDA approved as well. And they use EZH2 inhibitor either in itself or in combination with different inhibitors for very specific indications or specific tumor subtypes. We also recently, like a couple of years back, showed that there are different mutant tumors become sensitive to EZH2 inhibitor. There was a paper in ovarian cancer where there is ARID1A mutation. Mutant tumors become highly sensitive to EZH2 inhibition. So we had a cohort of bladder cancers. We found that around 25% of bladder cancers have this mutation, ARID1A mutation. And we did a lot of experiment. The simple result basically is that if there is a mutation in this ARID1A gene, they become super sensitive to EZH2 inhibition. This is the mouse model. This is the patient-derived xenograft model with the mutation of ARID1A or without. This is basically a concept called synthetic lethal interactions, basically. ARID1A is a complex that activates genes, and EZH2 is a complex that inhibits genes. So we are now targeting that inactive ARID1A patients with EZH2 blocking. And based on this, a couple of oncologists are setting up clinical trials for bladder cancer patients with this specific mutation with EZH2 inhibitors now. Of course, EZH2 is one example. We worked extensively on ERG as well, and several other biomarkers. Most of the time, we in the group picked up enzymes as a target to go after, purely because enzymes are slightly easier to target than, let's say, transcription factors. Some of them, like I said, EZH2 has a FDA-approved inhibitor. Some of them are drugs that is available for some other indications, like, for example, losartan is used for hypertension. But we see around 10% of breast cancer have AGTR amplification like that. So some of the targets I mentioned in this slide have experimental drugs now, and hopefully, in some years, some of them will become, available as FDA-approved drugs. Particularly, I am hopeful on this gene called PAICS. It is an enzyme involved in de novo purine biosynthesis. And we all know, the folate inhibitors like methotrexate has been used in some cancers from last 70 years. Dr. Subbarao, Yallapragada Subbarao developed those inhibitors. But this enzyme is more specific, and the de novo pathways are used mainly in cancers compared to salvage pathway that is used in the normal cells. So hopefully some of these targets will have drugs that are available soon. So just to summarize, we recently developed the UALCAN platform so that everybody can look at all the gene expression or proteomic data to identify certain targets in specific cancers or specific type of cancers. We worked on several targets like EZH2 and understood the biology of genes like EZH2, where it acts as a master regulator of gene expression. And now, of course, some of them have FDA-approved inhibitors. So that is the our research from last 25 years. And of course, a lot of people helped develop all these tools like computational tools, because I'm not a computer scientist, but we wanted to show this data in specific format that these people helped. Darshan and Chad were involved in UALCAN development, and others like Jed Ferguson was involved in ARID1A mutant bladder cancer studies. A lot of work at Michigan was helped by Arul and Nalla Sivam, as well as Selena Clear. And some of the funding support came from NIH and DOD as well. With that, I would like to thank again for this opportunity. And if there is any question, please let me know, or you can always email me. Thank you.
Thank you, Surya. That was a wonderful talk. Very useful tool that you guys have developed. So I think it's. I would expect it to be more popular than what you have shown. How old is this? How long ago this was developed and released to the public?
This is 7-year-old. So
it's
still people are using it. And one important thing is we want to keep adding new data sets so that people come back and use it. Yeah.
That was my another question. How frequently update this data?
That's what, one of the important update we do is the patient survival information, because NCI updates it every 6 months once. So we'll know how many patients died or still surviving, those kind of information. And then, clinical proteomics, they are increasing the patient samples. And when they release, we want to, include those data as well. One of the, I am eagerly waiting is for the Indian cancer patient samples data.
Okay.
So the ICGB in Delhi, they said they have 300 breast cancer patient data, but it involves a lot of paperwork, I think, for us to receive the data. And then, like I mentioned, they started a consortium called Indian Cancer Genome Atlas. They want to replicate what the Cancer Genome Atlas of NCI did. So their initial goal is breast cancer sequencing, and they're proceeding with that. And once the data comes, we'll include here as well.
Do you know, is there any reason for focusing breast cancer in India? Because India's lung cancer is the most popular, if I understand.
I think they had more advocates going for breast cancer study. And I think breast cancer is also pretty common in India. Yeah. You would think maybe head and neck or oral cancers because of tobacco chewing and all that. It's more common. But yeah, they went with breast cancer actually. I mean, ultimately,
I have another question. When you compare the upregulation or downregulation at the genomic level with the protein expression, what is the correlation?
That's what many people did, including we did. It was a cancer cell paper actually. We did correlate individual patients' protein and RNA level. So we saw around 75% concordance. That is, if the gene is overexpressed, protein is also overexpressed. Around 25 did not behave like that.
Okay.
So it went opposite way. And we can always explain with different reasons, protein stability or post-translational modification, degradation of proteins, things like that. So, but it's not 100%. If there is gene overexpression, it's not always protein is overexpressed. Yeah.
35% is still a good correlation.
Yeah, concordance. Yeah. I think Dr. Karnik has a question.
Oh, yeah, I want Ramesh to finish.
Yeah, sure.
Go ahead.
Suri, very nice talk. Although I'm not a cancer researcher, occasionally I visit your site because particularly interested in gene expression, and because there is so much overlap between vascular system and the cancer system, I think we find your tool to be extremely useful to us as well. Because you started now making it available on your iPhone or Android,
Yeah.
I'm assuming that it's not the NIH reviewer who is looking at your site using the iPhone. It's actually, patient relatives in distress. Okay. I have always been contacted by relatives, friends of friends from India asking me if there are NIH clinical trials going on in this particular cancer. Can you put us in touch with someone who knows these things? So I think that is where this tool is going to be extremely or probably highly used in that type of users.
Yeah.
What I found that if you are attracting attention from the distressed family members, your tool should actually also start incorporating some way for them to guide to either clinical trials or potential experimental drugs available for targets that may be unique to that particular patient's cancer. I was wondering if there are efforts or efforts to come to link your database to the International Union of Pharmacology database, where both experimental as well as FDA approved drugs are listed for every possible protein in the proteome of humans.
Right. Absolutely.
So
Yeah. Yeah, that's a very, very important point. And actually, we do have actually any gene you take, if there is any drugs available, not the FDA approved, it shows a link. In the external links, we have something called drug bank. So idea of putting that drug bank is also for even researchers to repurpose that. Let us say losartan, for example, right?
Yeah,
Cardiac.
exactly.
Yeah, that kind of cross, or it's called drug repurposing, right? We do have that. And the other thing you mentioned, you all can definitely helps even other type of researchers, because like cardiotoxicity of cancer drugs is one of the biggest area of studies. I think one of the big areas of studies. So that is where you can potentially look at different genes, what is going on, and then see maybe those genes are responsible for cardiac toxicity or things like that. But definitely the drugs that are available, we do provide for any of the targets, and that can be potentially used by the families, and then contact the oncologist, say this is there is drug available, particularly FDA approved, because the experimental drugs nobody can take, right? Even though the patient's family says, hey, there is a drug that shows up here, but oncologists can't use that. Yeah.
Sure.
But they can participate in some of the clinical trials.
Right.
Based on what I know, it is actually access to the clinical trial itself that patients would be interested in.
Yeah.
Especially cancer patients with new targets.
Yeah, right. That's true. And then the other thing is the time lag. In India, I think still a long way off with this precision medicine. And one of my friend's dad had a specific mutation, mutated lung cancer. He used to get drug from Taiwan, or I think Taiwan. It was costing around 8 lakhs for 6 months dose. And he survived with that drug for like 5 years or so. That drug was not available in India for any purpose. Yeah.
It's very nice. I'm Srivatsan, because we work on cancer too. I mean, I work with HPV and
Cervical.
basic science, of course, cervical cancer and head and neck cancer, both for diagnosis as well as for the therapeutics. And it's interesting because EZH2, one of my colleagues, is an oncologist, pulmonologist. He works EZH2 alone will not do it.
Yeah.
You do it with the immune repressors, immune suppressors,
Right.
anti-PD-1, anti-PDL1. They've been doing it because prostate cancer is a big one at UCLA. So the top centers of like you said, Michigan, MD Anderson as well as UCLA, they are very good in top notch in prostate cancer. Still, the major problem always been resistance, because you were mentioning about the purine. I just reviewed a paper because I didn't accept it. Pyrimidine pathway. So there are always pathways, metabolites, because instead of going to the genes and RNAs and transcripts, you go into metabolites. So if you can start the metabolites, like we said, the purine pathway or pyrimidine pathway, they control many of these ones, metabolites.
Right.
So it's interesting because people have been trying to do it. In our case, because like something I just thought I'll mention to you, one of the ubiquitin ligases, we said that's altered in head and neck cancer. A lot of things are there. And post-translational modifications, because you're talking about post-transcriptional modifications.
Yes.
But RNA, no protein, the protein is there, it doesn't complex. So it doesn't make a complex. So even the protein is there, even if you do the immunohistochemistry, protein is there, it's still not useful because it is not forming complex with other protein or by itself.
Right.
So complex, that's another one more layer of problem which is exhibited by these systems. And so the multi-targeting, precision oncology with the multi-targeting, one of the things we do, I mean, clinical sponsors paying, otherwise we won't do it. Like you said, it costs millions of dollars. Especially PhDs, you cannot do anything. So sponsors, because we have a project for the turmeric. It comes in the form of lasagna, and it's very good. Phase 1 we finished long time ago, we published it. Now we are doing phase 2. Phase 2 has got we do the same thing. Yeah. The circulating tumor and many of those things. The RNA, mRNA, RNA, proteomics, everything else. I mean, everybody does the same thing. We do the same thing with UCLA and the VA here in Los Angeles. So hundreds of around 25 samples. a lot of money it costs to do many of those ones, multiplex and everything else. So it's interesting. So I think the multi-targeting, so as I gave you the examples of two things, ACH2 plus immune suppressors. Because head and neck cancer is one of the things we are interested in. Like I told you, I came from HPV.
Yeah.
So for diagnosis, not only we use the tumor tissues, we also use saliva, we use plasma, serum, PBMCs, a lot of different things we use for our analysis. Saliva is very good because we can look at HPVs. We collaborate with UPenn. So to see, they do different things and microbes, we do different things with respect to HPVs. So multi-targeting, multi-use of these various markers, because when KRAS is known for what, 1981, am I right?
Yeah.
Weinberg's business, they all find out RVRAS and then KRAS, no target is there. That's where the paper I was reviewing. It takes, KRAS you cannot target. I mean, after so many years, almost like 40 years, 41 years.
Yeah. And the pathway, other genes in the pathway.
So that's why you have to, from other way around, that's why you said the pyrimidine pathway.
Yeah.
So the precision medicine doesn't work precisely the way you want it.
Yeah.
It does have these resistance problems and everything else.
So the next level basically is, as you already hear, is the bringing AI to this, right? Where, like you said, multi-targeting, where probably you may reduce the dose of any of these drugs. The resistance comes with the drug dose as well. Let's say
toxicity is the problem, non-toxic drugs.
1/10 or 1/100th of the dose, but with combination maybe like 3 or 4 targets and drugs, you may reduce the chances of mutations, and that's where the AI in medicine is going, particularly cancer medicine, where they want to get all the information available from individual patients and then start looking at potential combination they can use. You are right. Padmini Sharma published a paper about immune modulation with these ES2 inhibitors and all that. Talking about HPV, we have HPV positive and HPV negative head and neck cancer and cervical cancer information in UALCAN as well.
Yeah, it's a problem because you were talking about, yesterday I was there when some of the people were talking about UCLA, they're developing AI for head and neck cancer, looking at it. I asked the question, they don't know it, because it cannot differentiate between the inflammatory cells and the cancer. So you have inflammation after you eat some pickle or something, you have an inflammation in the tongue. 10 days later, you don't know if the cancer is the inflammation. So it's a problem. So there is AI, unless you train it, because AI is not something else.
Sure. Yeah.
Non-human.
It takes time.
You have to train it.
Yeah.
So when you train it for the cancer business with hundreds of cancers, it doesn't differentiate inflammatory cells. They are presenting it yesterday. I asked a simple question. They said, it's a good question. We don't know yet. So
it takes time. Like I showed a slide with the new targeted therapies. There are a lot of new drugs are coming, that can be used for specific subset of patients. So only hope is there. And ultimately, the cancer is one of the biggest cause for cancer is the age. So
I think it's age related. I think that's a major point. Age related. People live 90 years and then get a prostate cancer. What do you do? So, so you want to treat them, not treat them? So no idea.
Right.
Thank you very much. It's a wonderful seminar. It's beautiful.
From the AI point of view, this is really a gold mine. Such a comprehensive list of things. So
right.
That's right.
No, AI you can only train for, not for molecules. You train for the.
But for the training, anything, you need a good set of comprehensive data, right?
Yeah.
So this is the beginning. This gives a very good picture of, at, gene level, RNA level, and proteome level, and also it links to the cancer patients and whatever the progress in the therapy outcome. So in that perspective, it's a very good set of data. And it may not be comprehensive, but it's the beginning of such a valuable tool.
And other thing I must add, earlier, maybe 20 years back, if I was working in prostate cancer, if I tried lung cancer, they would kill me. Like, you are not expert. But now it's molecules, right? There are some common molecules across cancers, whereas there will be very specific. ERG is only specific for prostate cancer, whereas EZH2 is across cancer. Things like that. Knowledge is developed over the years. And probably, in 10 years, maybe we'll be looking at different aspects of, particularly I'm thinking about reduced dose and more new targets with FDA approved drugs. those two combination may be very helpful, I think.
Yeah, like I said, turmeric, I explained to you, we got a lot of different, I published a lot of papers on those stuff, on the curcumin and other additives, different pathways. Maybe TNF pathway or NF pathway, or like the one you were talking about, EZH pathways.
Yeah.
So, because that's one of the things we always try to do as basic scientists, trying to find out what the resistance is,
Right.
cisplatin resistance, why, how we can overcome it.
Right.
And curcumin can, turmeric can overcome cisplatin resistance. In vitro, and of course, mouse and humans, we are showing in humans too. We can take cisplatin resistant and treat them. They all want it. So it's interesting. Inflammatory and anti-inflammatory and anti-carcinogenic. Turmeric is very good.
And other important thing is we must add the mRNA vaccine strategy. They're looking at very potential positivity there, as well as the immune therapy, different immune therapy.
Immune therapy again works very well for some of these ones, like
Right.
specific one, BRAF mutation business, melanoma. So it works very well. Including Carter, am I right? His brain cancer went away when they did this anti-PD-1 inhibitors. But then you go to lung cancer, it doesn't work.
Yeah.
So you need to add something and something else.
Right.
So melanoma is very good. Okay, it's a wonderful drug. Okay, Rebus is there. Tony Rebus at UCLA, one of the first ones. Of course, the Merck drug, anti-PD-1 or anti-PD-L1. It's very good. The melanoma goes away. Like we said, Carter, 5 years ago he was treated. He doesn't have any tumor. He's aging, 98 or whatever it is. His tumor went away. Brain cancer went away because it's metastatic melanoma. So again, the targets, even if you have the same target, BRAF mutation, whatever it's called, 603 or whatever, still it works in melanoma. It doesn't work in lung cancer or the one I reviewed, some other cancer.
Yeah,
So
unlike flu or fever, cancer, one size doesn't fit all, right?
That's a problem. That's been the major problem here. I think that's where the precision medicine is also learning. And then we'll wait and see what the AA does. And it'll be nice.
Also, it's sometimes it becomes unrealistic. The cost of some of these
Cost.
only rich countries and rich patients can afford, I think.
Yeah, there was an article in Nature yesterday or last week. I forwarded a lot of people. They talked about this CRISPR.
CRISPR in Delhi, right? The patient died.
CRISPR in Delhi, and then Bangalore one.
Yeah.
One in the Bangalore. It costs around $3 million per year for the because there's a down cone. He has been doing this for the sickle cell anemia, single mutation. Because we did it, polymorphism, single nucleotide polymorphism to convert it. So it's difficult. Even cell line model is so difficult. And you can imagine the human model, humans, people, around $3 million. So this fellow is very good. This is Ghosh and Chakrabarti and others.
Yeah.
Unbelievable. Rare disease.
Yeah.
It's so disease. You got everybody in UCSF, they've been doing it, Doudna and others.
Yeah.
Still, it's so difficult. Because a rare disease, it costs $3 million. Per dose, what do you do? And you don't know the toxicity. It's interesting. In India, they are following it up. In Bangalore, that fellow is very excited. That Ghosh, he wants to make sure it's available for everybody at a cheaper rate. He says we cannot be paying $2 million for a dose.
Any of that costly things, if you do it in India, it will become cheaper for sure. Because
So he says he wants to bring it down to $100, which is very good. If you can do a CRISPR modifications for sickle cell, that would be great. $100. There are centers are available. So I think it's various models, AI model, CRISPR model, multiplex model, multiple treatment, plant-based models. Models are available. We don't know which one works. And people are living longer. So that's, take it. People are living longer. So 80, 90 has become common. Thank you very much. I'm sorry I took away too much of your time.
I had a quick question. Is there any insight that one can generate regarding the cell types that are likely to mutate in these different cancers? Are they actively dividing cells? Are they not getting proper nutrition in terms of balance of metabolites? Is there any conclusion that one can draw based on the studies that you have done as to where and when these mutations occur? And also, I was curious from a spatiotemporal perspective. You showed a very simple diagram of a breast cancer I think, how it actually became a metastatic cancer,
Yeah.
but, or maybe it was prostate, I don't remember.
Yeah.
But be that as may, are there insights that one can get regarding the cell types that one needs to focus on in terms of nutrition, and especially with the imaging capabilities, can one actually make sure that those cells are targeted for healthy versus somewhat metabolically perturbed cell types?
Yeah, I
So
mean,
that's what I mentioned about the AI.
Yeah.
Yesterday's talk, the head and neck cancer. They are looking at the metabolic activity, the flux. Metabolic flux is what the AI picks up.
Right.
So it picks up and then says some specific cells within that area in tongue, the head and neck cancer, the
Right.
tongue. There's a metabolic flux. So the AI is able to pick it up and puts it like a IR map,
Right.
irradiation map. So yeah,
Right.
here is so they can surgeon can go there and take out those cells. So you are absolutely right. It's three-dimensional. You can
Yeah.
rotate around.
there
So
there
that's
are
what the
many.
AI is trying to
The
do.
advancement is in different areas. The radiology, the technology itself, the surgery. They have the robotic surgery going on. So as per cell types itself, the old hypothesis still holds good. They are doing all type of experiment that there will be always mutations happening in our cells, and there will be repair or cell death, program cell death. But there will be some mutations that, in fact, Bert Ogleston even used the word, it's bad luck, okay?
Bad luck.
That is the situation where some mutation somehow makes some cells become cancerous. Otherwise, all mutated cells, if you sequence, let us say, assume my lung is normal, you do find mutations. That doesn't mean it will cause cancer. So there is the second hit hypothesis by Knudsen, where they think there will be one mutation is not enough to cause cancer. There will be additional insert. And the biggest study is very important, I think, is that the pollution, that was a Nature paper last year, where they looked at the EGFR mutant patient, lung cancer patients. The second hit is basically pollution. That is the crux of that paper. In big cities in Taiwan, I think, and China, they measured the cancer incidence along with EGFR mutation. They found that the pollution is a second factor. So, I mean, it's scary, but it's a fact. So there are many hypotheses where they think that may be the reason, but ultimately could be bad luck too, a little bit right now.
Well, I mean, anything that is pro-inflammatory will certainly have that kind of an impact. So it's not surprising that polluted cities, which cause all kinds of inflammation, everybody looks at the lung, but there is no reason why eye tissue or the tongue tissue or some of the other exposed to skin tissues, etc.
Bladder also.
Yeah, bladder. I mean, this has to be beyond what is generally thought of as being exposed. But I was also interested in how the anti-inflammatory molecules, because I think we just had a comment on turmeric, and turmeric is just one class of molecules, but we have other molecules. I was wondering if we can actually begin to segment people who have this preponderance for not repairing the DNA, and then those who are able to repair the DNA and the cell types that are typically affected, then we can actually come up with preventative mechanisms which are going to be vastly superior, and that might actually become a part of the therapeutic management, because that we are missing the dietary components in many of these diseases. Yeah, people have started trying. Yeah.
They always prescribe Mediterranean diet as the healthy diet. Definitely some of these things we use from like centuries, the turmeric and even tamarind, you name it. They're all probably useful components as a preventative. Yeah.
Kishore, when you're talking about cure in sickle cell anemia, I just gave you an example. Thalassemias, single mutation is known for ages. Still difficult to treat them, except for maybe
there are 4 patients now. There are 4
pediatric.
patients.
You can activate that the blood hemoglobin A or something. Then it's okay for 10% of people. So it's not easy. I mean, targeting single cells or
Right.
few cells, it's not going to be easy. It's age related. People will give longer and longer, and some other disease will come around. So
Well,
not
well,
like
actually,
Mm, yes.
one of the things that has happened, Srivatsan, is that people are also looking at the amino acid transporters and peptide transporters and are looking at starvation of the tumors, right?
Got it. Yeah, starvation.
So the beauty of these data banks is you don't necessarily have to put a hammer to the target.
You're right. Yeah.
But there are probably some jeweler's screwdriver type of tuning that we can do and kill the tumor cells. The fundamental issue has been understanding the stem cells and the key stromal cells which are responsible, and then knocking them out. But to the extent that one can get sophisticated with these kinds of spatial transcriptomic data and temporal transcriptomic data, one can actually induce cell death, which to me actually is going to play a more significant role. One doesn't have to think about this as $1 million problems or $1 billion problems. One could also, and this is the beauty of going to a place like India, one could begin to experiment in a different way than the traditional therapeutic approach.
That's what we are trying to do. See, like saliva, we are trying to do saliva, HPV, and few other microbes, everything else. So we are developing some of those ones. And if it is useful, it'll be very easy. Any of the villages we can go, collect thousands of this saliva, analyze them. It's very easy to analyze in PCR and whatever the methods we are using. You can analyze them and then find out who might be getting it. Because like we always said, HPV, you and I and everybody will have HPVs and other things. We don't develop cancer.
Right.
Like you were saying, it's a bad luck. Some mutation comes along, somebody develops head and neck cancer or whatever the cancer.
Yeah, talking about India, one thing, I'm adjunct faculty with KMC Manipal and Mangalore. I always discuss with them. One of the important thing I am trying to tell them to initiate is developing a biorepository, right? They don't have any properly annotated biorepository to go back and check for any of these events that you are talking about,